I would just like to welcome you all here for this evening's BMS Talk. We are delighted to welcome Dr. Carolina Coelho from the University of Exeter, who is going to be giving us the talk this evening. I'll just run you through what will happen. So Carolina will give us this presentation. If during the presentation you think of any questions you'd like to ask Carolina, can you just please pop them in the chat, and then I'll come to them at the end of the meeting, and then we can discuss the questions at the end. So my name is Jan Quinn. So I am a medical mycologist. Like Carolina, I'm also currently the General Secretary of the British Mycological Society. But it gives me great pleasure to welcome Carolina here today. So Carolina is a medical mycologist. She has a long-standing interest in the human fungal pathogen Cryptococcus neoformans. She's done a PhD and then postdoctorate work studying this important human fungal pathogen, where she was with Arturo Casadevall at the Albert Einstein College of Medicine in Bronx. So she was in the US of A. And now she is a group leader and lecturer at the MRC Centre for Medical Mycology, and she's been there since 2019. Carolina was also a recipient of a BMS research grant, and so it's with great pleasure that we welcome Carolina here to talk to us today about the great work that she's doing on Cryptococcus neoformans. Okay, Carolina, I will stop my video and mute myself, and I'll let you take the stage. Thank you very much.
Thank you, Jan. Thank you, BMS, for the invitation to give this talk. It's really an honor to give one of these talks. And thank you, of course. I'd like to appreciate the grant support, but also all the rest of the work that the BMS does for the community. It has been really interesting talks, sometimes with mushroom walks. So really great community. I'm really happy to have known this community in 2019 when I came into the UK. So with that, let me take you to my talk. We'll be discussing Cryptococcus neoformans infection, how Cryptococcus neoformans invades, and the innate immunity to Cryptococcus neoformans. Okay, so for the purpose of this talk, I would just like to introduce the fungal pathogen Cryptococcus neoformans. And actually, one interesting detail about this pathogen is that Cryptococcus is very distantly related to all the other fungi that cause disease in humans, and it's actually one of the closest ancestors or related families to Cryptococcus neoformans is the bolero mushroom that we eat when we can, also known as porcini or penny bun mushrooms. So it's actually really related to Cryptococcus neoformans. Cryptococcus can have, let's say, a multicellular form. The form that we encounter in the host, and we see this in microscopy, is this very round yeast, and it can have varying sizes of the yeast, but it tends to be relatively round compared to other fungal pathogens. It doesn't form hyphae, not during infection. It hasn't been detected during infection to form hyphae. So in that, a couple of unusual characteristics of Cryptococcus neoformans. And then there's a really unusual characteristic that you see here. And let me just double check that you can see my laser pointer here. A really unusual characteristic that you see here is that it has this halo around the yeast. So this would be the yeast forming this halo. And this is what we call a polysaccharide capsule, because it's polysaccharides made of sugars and it's completely transparent. So it's also here in this picture, but it's just transparent to light, so you can't see it when you have a microscopic slide. But when you darken the background with India ink, then you can see the capsule, because the India ink just pushes against the capsule. You can see this halo of the capsule. And this capsule is basically an invisibility cloak, because these sugars are poorly recognized by the immune system compared to other cell surfaces that we use to start fungi. And it really provides a couple of very unique characteristics to this fungi when it's causing disease in the host. We know a little bit about the capsule, but we always highlight this because it's such a unique characteristic of this fungi compared to other fungal pathogens. So Cryptococcus is mostly associated, and it came to notoriety during the AIDS pandemic. So these are the statistics of cryptococcal-associated deaths. And even in 2022, Cryptococcus neoformans and related species that are included in this pathogenic complex cause 15% of AIDS-associated deaths worldwide to this day, right? And that's over 100,000 deaths each year. And even in the clinical trials with the best medical care, even in the Western world with the best medical care, there's still 20 or 30% mortality to this pathogen because it's incredibly hard to treat once you have the disease installed in your brain. And to really illustrate that, I show you here an MRI scan of a brain. This is a normal brain, kind of the oblong shape of a normal human brain. When Cryptococcus is growing, can really invade and grows to such high amounts in the brain that actually all of this is fungus, and it's kind of dislodging the central ventricle of the brain, the central septum of the brain. So you can imagine this would be the MRI of a head of the person who's seriously sick, and it would be really difficult to survive without sequela in this patient. So really dramatic disease, let's say, even though it's mostly associated with HIV. The funny thing, again, a little bit different from a lot of other pathogens, is that animals get sick too. So I highlight here horses, cats, any sort of like cats in zoos, monkeys in zoos have been known to get Cryptococcus. Dolphins have been reported to get Cryptococcosis. And a really prevalent case for those of you who like veterinary pathogens is actually Cryptococcus is thought to associate with trees, particularly eucalyptus trees, the species Cryptococcus gatii. And this causes infections in koalas because they're there in eucalyptus trees all the time, eating the leaves. So there's a couple or a group of veterinarians in Australia who have really great work in veterinary cryptococcosis, but koalas is a particular example of an animal who's really affected by cryptococcosis. So with this, I've shown you the image of the brain. I've shown you how Cryptococcus looks when it's in the laboratory. And here I'm also showing you how badly infected a lung can be. So this is the outline of a whole lung of an animal, and every little blue dot is Cryptococcus neoformans. So it can really grow to huge amounts in the lung. Every little dot, and here is the magnification under the microscope, can be Cryptococcus neoformans. So it's really, really high loads causing disease in ammonia in a human or in animals. And why does this happen? We know that Cryptococcus is in many trees, particularly, or it's very more frequently found in eucalyptus trees. It's also frequently isolated from birds and bird guano, so it passes through the digestive tract of birds. And then what is thought is that from these environmental reservoirs, either spores or desiccated yeasts of Cryptococcus neoformans, of Cryptococcus species, will go into the air, become airborne, and they get inhaled into our airways. And they can lodge into our airways, go into our lungs, and then they may, at that point, undetermined, disseminate to the brain in humans. But usually, this is a process that causes no problem in an immune competent host. So if you're healthy, this will not cause any problems. The vast majority of us have already been exposed to Cryptococcus neoformans, and it doesn't cause a problem until, this is what we highlight here, there's an immunosuppression. And in immunosuppression, the immune system cannot fight Cryptococcus neoformans anymore, and you start having brain and lung disease, and this is life-threatening, right? And the thing that I find interesting from a historical perspective is when the AIDS epidemic came, we understood that because the AIDS epidemic leads to a loss of the CD4 positive T cells due to the HIV virus, this leads to a depletion of CD4 T cells. And we think that because the CD4 T cells help other innate immune cells called macrophages kill the fungus, the loss of the CD4 T cells influences this killing axis. And that's why AIDS is so associated with Cryptococcus neoformans. And this was kind of like known from the 80s. And as years go by, and we're now in 2025, so decades later, we still have the same image but we have a lot more detail on it. So now we know that one of the molecules that's critical to communicate between the CD4 T cells and the macrophages is interferon. So any genetic variants of interferon gamma or any autoantibodies against interferon gamma can lead to increased susceptibility to cryptococcosis. The same happens for the cytokine TNF alpha, phagocytic receptors, and signaling molecules, which we're not going to get into, signaling molecules that are involved in responding to these cytokines. A loss of these cascades will basically increase susceptibility to Cryptococcus neoformans. And there's other molecules that are also kind of fitting into this innate immune killing directed by T cells and a little bit by B cells via antibodies. So we have a picture based on the association with AIDS, with a live disease. And now we have a lot more detail in the past decades. We've gathered more detail, but we still fit into this picture. And in order to illustrate, since this is a talk with the broad audience, in order to illustrate this, I'm going to show you a video that I tend to show in my classes, if it works. No, it's not going to work. No. So I'm not going to show you a video of this class, but this would be a video illustrating killing of phagocytosis. But I encourage you to follow this link if you have the opportunity. Maybe we can put it on the information, because this is a really funny video, a really old one, showing how killing can exist within cells, how cells can kill other cells. Oh, there you go. So this is an amoeba, and this illustrates phagocytic killing. So a macrophage is derived the name macro from big phage, so big eater. And what you see here is an amoeba doing a macrophage activity. So it has ingested a paramecium, and now you see how this enclosure is really, really uncomfortable for the paramecium. The paramecium is trying to escape, and you'll now see it stopping moving because this ingestion of this big eating, this process, happens all throughout the tree of life. And in our immune system, it's what helps innate immune cells to kill pathogens. And it's a really important process to have this CD4 T cell macrophage killing of the fungi is a little bit like this, analogous to this. So here we have the recapitulation again. And this is kind of the general immunity to Cryptococcus neoformans. The interesting thing is we know very little about this very first stage of invasion and dissemination between the brain. So in this step here, where Cryptococcus is inhaled into our body, lodges into the airways, possibly goes into the brain at an early stage, we know very, very little about this stage, both in humans and in mouse. It's a stage that we don't really know. We know that in the lung, there's an innate immune system and an immune system, and then it goes through. Cryptococcus has to reach the brain, needs to cross the bloodstream, needs to get disseminated through the bloodstream into the brain, because those are distant sites. And all these stages, the immune system from the lung, from the bloodstream, is going to try to prevent this trip of Cryptococcus neoformans. And we know, or we're starting to characterize now, how Cryptococcus can do this invasion, but it was still really early days. But we know that Cryptococcus has the capsule to help disguise and to be an invisibility cloak for the immune system. But we also know that it secretes a lot of immunomodulatory molecules, or to avoid the fungal killing or to disrupt cascades that activate the immune system, also proteases for tissue breakdown. We're starting to have a picture of how this happens, but it's still a lot to be discovered. And in particular, when we reach the brain is well protected by a blood-brain barrier, which is an extra separation between the bloodstream and the actual tissue. And in the brain, and we don't have to know this, but this would be a diagram, the blood-brain barrier, this extra protection, is made of endothelium, the vascular wall, the wall of the blood vessels, and then at least 3 other different cell types, parasite, astrocytes, and in particular, the microglia, which are the innate immune system cells of the brain, and that they are also called brain-resident macrophages. So these are the cells who are monitoring the blood vessels and trying to protect the brain and giving the brain this, the blood-brain barrier, this particular capacity to isolate drugs and to be protected from the outside. And if you work in neurodegeneration or any other neurological diseases, you know that there's a problem in getting some drugs to reach the brain. And part of it is because of this increased protective barrier of the blood-brain barrier. And we need to overcome that. So what do we know? Starting from the bloodstream, the first thing we needed to study this, how fast we needed to do a kinetic. Starting from the bloodstream, how fast can cryptococci reach the brain? And work a while ago in 2013 did this by injecting Cryptococcus straight into the bloodstream of mice. And what they did is that they discovered that they could start detecting, if you inject Cryptococcus into the bloodstream of mice, at 24 hours post-infection, you can already detect fungi within the brain. And then the fungal load is increasing further and further as days go by. And in 4 days, it's very easily detectable. And you start to have cryptococcal masses here via microscopy. But this is starting from the bloodstream. But the issue is that we think that the route of exposure is actually starting from the nose through the lungs. So starting from the lungs, how fast can cryptococci reach the brain? So in order to answer this, we took some mice and we performed intranasal infection because that is the model that we currently have to mimic nasal infection. And after trying several different time points, we found that we could always find cryptococci in the brain. And at some point, we actually went as low as 3 hours and 24 hours. And we imaged because we were looking for the very early time points of invasion. We did CFUs as but we also did imaging to demonstrate that it was Cryptococcus. And I show you here to microscopy images, and it's hard to see because there's not a lot of crypto, but this green dot here is crypto well inside the brain tissue. And here is a cryptococci in the edge of the brain, but definitely already traversing into the brain and already well situated into the brain. So we already know that with this experiment and others, we already know that there's an extremely fast brain invasion after an intranasal infection. And this tells you that actually these mechanisms of going from the lung through the bloodstream and then through the blood-brain barrier to get into the brain are actually really sophisticated because they can be so fast, right? So there was a couple of clues here, and there was many different ways of studying this, but we had to take a breather here and figure out how we were going to get different information and what was the best tool to study this. And I always use this cartoon because this cartoon kind of shows you the different perspectives, the different routes you can have on the same problem. And by studying it from different angles, you can have different answers. But if you don't forget to look at all the different angles and the work from other people, you can start building an actual picture of what's going on. And that's always been a really big concern, trying to integrate the different data from all the different techniques. So I talk about this because there's two theories about how Cryptococcus can traverse from the bloodstream into the brain and how it goes from the lungs into the brain. And one is that from some way, free yeasts, free cryptococci, go into the bloodstream, and when they reach the blood-brain barrier, they rest and they traverse the blood-brain barrier, right? And then they can start interaction with some immune cells of the brain, microglia. But there's also been some suggestions that there is another mechanism where Cryptococcus inside innate immune cells, the macrophages, from the lung, because they want to potentially because they want to transport the yeast through the lymph nodes, then the macrophages will carry the yeast through the lymph nodes. And then in some way, instead of arresting in the lymph nodes, as it would happen if this was an effective response, it continues to traverse from the lymph nodes into the bloodstream, and from there it reaches the brain. And actually, then it could have a Trojan horse mechanism where yeast could traverse the blood-brain barrier and other organs via Trojan horse mechanism, where it would be carried inside a phagocytic cell, which can traverse the blood-brain barrier and other vasculatures. And there was this two competing hypotheses, and it wasn't really clear which one was prevalent, if one was just incorrect, or mostly if which one was prevalent, and how do they vary? How do they ratio vary, right? So, for example, free yeast or Trojan horse, the debate of these questions, Santiago Tirado and others have been working on this. And in vitro, they have a model of a vasculature. And when they have the model of the vasculature, they can put it on a layer and they have what we call a chamber. And you can separate the two sides of this chamber by putting a blood vessel or a blood vessel mimic model, right? And if the free yeast can traverse this model of brain-brain barrier, or whether they quantify directly whether the Trojan horse yeast, so the yeasts in red, and then inside a phagocyte, could traverse the endothelial barrier that they created. And then did this in a model of cell culture with human endothelial cells, right? And then, of course, they quantify these either by CFUs or via imaging. And what they found was that Trojan horse is actually possible, but actually, it's also possible for the free yeast to be traversing. So they seem to really suggest that both traversal mechanisms can happen, but there's still a question of which one is prevalent, what happens in vivo, right? And we thought about a really, really long time about how to answer this question, because if you really want to study invasion very early on, what occurs is that you have to study rare events. And in order to study rare events, that makes it really hard to quantify, and really hard just to detect them, right? So we had to think about a really long time. And then there was a couple of advances in this technique that I'll tell you about, but we chose a particular technique that we had to implement from scratch in the laboratory, and that I really think that it's going to be a technique that we can use for many, many different problems. So if you have a research basis, I really recommend you try this technique, and I'll explain why. So this is using what we call tissue clarification. And tissue clarification, in a nutshell, is we are opaque, light doesn't traverse our bodies, it doesn't traverse the body of animals. But as early as 1905, so really past century type of things, there was attempts and there was some progress made into trying to make tissue less opaque. And how do they do that? Actually, tissue opacity is due to light scattering because of the different refractive index between our skin and the lipids that we are made, and all the water and lipid layers that you traverse, right? And is also given by light absorption, pigments such as heme or melanin in our body that gives pigment, and then they absorb light. So the principle between tissue clarification is that if you homogenize the refractive index, and you do this by removing the lipids, so it's kind of a little bit like removing fat from milk, and then it becomes less opaque. So cream is a little bit more opaque than very, very, very skim milk, right? And then also by removing the pigments, right? And then the tissue becomes more transparent. And in the early 2000s, this really had a big jump start. And a couple of researchers really, really were able to get this going in a very practical way. And I show you here, this is a mouse, and I can show you how after the clarification, this body is completely opaque. And after clarification, the entire body becomes transparent, so much that you can see the grid behind the animal. And you can imagine now that then this allows you to study processes such as nerves, such as neuron communication in a really in-depth way, right? And you can see here, this is the same animal in a fluorescent microscope, and you can just see a lot of things in detail, and you can follow the entire path of the nerves, right? So all of a sudden, using tissue clarification, you don't have to do sections in histology, millions of 2 micron or 10 micron slices. You can do whole body imaging of animals at cellular and subcellular resolution. Again, you can do whole body, but you can also see all of the details. And This tendentially uses microscopes that are special because they have speed and a good optic capability, and this is light sheet microscopes, which is part of the reason that Betsy got the Nobel Prize a few years ago, right? It's the faster and a very capable microscope, right? So when we heard about this, we really were happy, and we thought that we could use this for studying a process that we needed to study almost entire organs. It was really rare events, so we really needed the capability to understand things at detail, but also at a high volume, because we didn't have that many events to start with, right? We needed that high capacity. So we set it up in the lab, and the experimental protocol is very easy. We do our intranasals, our intravenous infections, in order to study the dissemination. And then we extract the tissue when we fix it, and we do sagittal sectioning. So sagittal sectioning means cutting from the nose through the bottom of the head, and that was because that was the sectioning we wanted. And I will highlight here, we did not do imaging of entire organs, and that was because we didn't have access to this particularly amazing light sheet microscope that allowed us to do that. But what we found is that, and this is an important bit if you're doing research, and even for those at home, the tissue clarification improves the capacity of every microscope. So we were able to do much thicker sections than we tended to do. We were able to get huge amounts of data and getting really in-depth and good resolution subcellularly with really, really normal microscopes. So if you have a laboratory, you don't need lattice sheet microscopy or fancy microscopes to take advantage of clarification. It will improve the resolution and the depth of imaging you have. And then what you can see here is our slices also became more transparent. You can see here that you can now see the lines through the tissue in comparison. On the bottom, you can really clearly see a line through the tissue because the tissue is now more transparent. And this is after staining. We put a little dye here to study to see the nuclei, and you now start seeing patterns, right? And this is a little image of a brain, and just to see how small it was what we were looking at, but this was million times more than what we usually studied, right? And we also needed to do this for the lungs. Those are the two organs that we want to study in detail, right? And we also found that because of what we wanted to study, we really needed to decolorize, remove the pigments from the lung because it has a lot more blood, a lot more hemoglobin. So it needs that, really needs the depigmentation step or decolorization step to study the lung. But we could also get a really nice image of all the lung. You can kind of see here the brachial tree. If you have enough resolution in your image, you can kind of see here just with a regular phone camera the details of the structures. So continuing here, here, this is an image I always show. This is just a stereo microscope, which is basically a magnifying glass that we have in the lab, right? And after we stained with the dyes we wanted to use, so one dye to localize the fungi and another dye to see more details in the tissue, in this case is the lung, we put it under a magnifying glass that had the right laser on it, and you can immediately have a look of how many fungi you have into the tissue. And this is, again, almost to the naked eye, right, with the right light. So it clearly shows the lung filled with fungi after staining, because the tissue is so much transparent that it sees the stain you added to it, right? And this is what would be the microscopy equivalent of that in a lot more detail. So wide applications of this tissue clarification technique. It will enumerate the fungal cells. It has a lot of flexibility in analysis of equipment. You don't need specifically, it will increase the microscope capabilities of all the microscopes you have. And as usual, technical advances means that you can answer more or better research questions. Really important tool for us. So what we did is we started putting a lot of different dyes into the tissue. And here I show you a picture that I'm really proud of because it really shows the brain and the communication with the nose, with a lot of detail into the vasculature and the different cell types in the brain. And if you do any sort of histology with this image, you can immediately see how much is going on in this image. And this is because this is a 400 micron thick image of a skull showing the cribriform plate, the brain and the nose, right? So we wanted to study invasion. So we did our slices. we clarified them, and we did intranasal infection. And we quantified to see if we could find the Cryptococcus, again, in the brain after early stages of invasion, and what cells it was associated with. So we're trying to define this timeline in better detail, right? We already know it's in the brain after 24 hours after intranasal infection. What can we do? So what we found was that we could find Cryptococcus in the brain after 24 hours after intravenous infection, and we could replicate the data from our colleagues showing that it's already starting to have little replication niches. It seems like these cells are either they're in little bunches here, and we could find the Cryptococcus within the brain. When we looked at the lungs, what we found was that if you look at this image here, this is a staining for a blood vessel, and it's forming a tube, right? It's the blood vessel. We could find free yeast. There are no associated mammalian cells within this blood vessel. We could find free yeast starting to be in the tube of a lung vasculature. We found 2 instances of this. We looked really hard. This is a rare event, so it's a fast event, so it's going to be really hard to catch it, right? Well, we could find here that there was at least 2 instances where we can find yeast inside the vasculature. So we kind of have the smoking gun that the free yeast can be inside the vasculature. In this case, it's not really associated to any immune cell. Again, supported of free yeast being the predominant mechanism instead of a Trojan horse. Doesn't mean the Trojan horse does not occur, but the free yeast seems to be the most frequent, the most predominant. And this is an image showing just the tissue context of this vasculature, where, for example, in green, you have the bronchiole stained with this green epcam, where epcam stained with green with a lot of fungi in the alveoli, which is where they usually are. But from here, in some way, they managed to reach the vasculature, and they are now found in the vasculature. So we found the smoking gun of there is free yeast in the vasculature. But what happens when we want to get that sort of detail, when we wanted to characterize the cellular detail inside the blood-brain barrier? What we found was that clarification indeed was a really good way of clarifying the blood-brain barrier. You can see here the microglia are stained. The macrophage cells that are associated with blood-brain barrier, they're stained in yellow. The vasculature is stained in red. And you can see here that they tend to be associated very closely with the vasculature. And in some cases, there seems to be a microglia that's kind of touching 2 sides of a blood vessel or 2 different neighboring blood vessels. And that's kind of what we've been probably really associated with their role in maintaining the blood-brain barrier, right? So what we found is that we continue to do these stains, and what we found was that actually, when we stain for the blood vessels and we also stain for the microglia, the vast majority of times, the Cryptococcus C we find after 24 hours of intravenous infection is the Cryptococcus is no longer in the blood vessel of the brain, but instead it has already passed through the vasculature, and it's already now past the blood-brain barrier or inside the blood-brain barrier. But the microglia seem to be the first cell or the most predominant to associate with Cryptococcus. And here you see that because all of the Cryptococcus in blue are associated with red, which is the marker for the microglia. So it does seem that the traversal of the yeast is quickly followed or predominantly followed by close association with the microglia. We found some instances where the microglia wasn't associated, and we don't know which cells are associated with Cryptococcus here or if it's really free yeast, because we would have to stain for the cells here. There's no cell that we can identify here because we haven't stained for all the cells that could exist in this space, right? So we don't know which cell is associated here. You can see here the microglia close to the blood vessel ingesting Cryptococcus, several instances of that. So we quantified this, and because now we can finally get the numbers to start quantifying this, and we found that the vast majority of Cryptococcus, at least 60%, 50 to 60% to 90% of Cryptococcus are associated with the microglia. And are no longer associated with other cells. So the microglia seems to be predominantly at this early stage the cell that is mostly associated with Cryptococcus and is going to ingest it and perform that process or attempt to perform the killing process that I showed you on the video before. We found low to no association. This is the data that I'm not showing here, but I'll tell you what we found. We looked to, or we stained for other immune cells and circulating immune cells to try to stain for the Trojan horse, and we didn't really find association with the other immune cells except for the microglia, at least not the ones we stained for, right? We didn't really see that. And this is the first, or one of the first, mammalian model data showing that free yeast traversal is predominantly the main mechanism, because we can find free yeasts in the bloodstream, and then after they cross the bloodstream, they're associated with a cell type that is only on the inside of the tissue, right? And it closely associates with the tissue. So what are the consequences of this? It's known that the tissue-resident macrophages for the brain, they're called microglia. For the liver, for example, they're called Kupffer cells. They perform immune monitoring of the bloodstream, and they do this, right? They can uptake infiltrated particles. They can uptake even particles from the bloodstream. And what has been found is that if the particle that the tissue-resident macrophages uptake is inert, doesn't cause an immune response, and this was tested with beads, there's little immune response. However, if there's an immune activating particle, and in this particular study, they used a particle called immune complexes that's very strongly immune activated, the uptake of these particles, inflammatory particles by tissue-resident macrophages, leads to a strong immune response. And in this case, this was kidney, and immune complexes really led to an interferon-dependent recruitment of neutrophils, right? And this was really interesting, right? Because if we then look at this, it means that potentially these microglia can be very strongly activated and can be secreting and communicating with our immune cells in a really, really interesting way. And for example, if we look at their morphology is characteristic of a cell who's already activated. And we're starting to wonder whether this microglia cell here in white is starting to communicate with neighboring cells and calling immune cells. And now with this tool and with aid of other tools, we can start to dissect this mechanism, so we can start dissecting whether this is interferon dependent, TNF dependent, or any other sort of cytokine dependent, which cells are called in first. So really, really exciting developments. And we know that immune cells, again, let's look at the elephant from all the sides. We know that immune cells are going to be called in because we already know from Koff and Francis and many others that at 3, 5, 10 days after infection, there's a strong infiltration of immune responses, of inflammatory immune cells recruited from the bloodstream into the sites of invasion. So what we're trying to do is now fill out this timeline from as early as 24 hours to days later, figuring out how this one cell then triggers infiltration of huge amount of immune cells to try to kill this invader. And at the same time, how can crypto from one to a few cells continue to grow in a granuloma? And can we tip the scale? And once we know the details, can we tip the scale into the favor of surviving patients, or helping patients survive more and decreasing mortality rates? So in summary, and I'm getting to the end of my talk, there's a free yeast traversal that our data supportive that the predominant mechanism is free yeast traversal. Trojan horse seems to, in our model, be less predominant. In 24 hours, Cryptococcus neoformans can cross the bloodstream, cross the blood-brain barrier, and now it's mostly interacting with a macrophage, resident macrophage from the brain, which are microglia, or a type of microglia, and this is for those working in neuroscience. It could be border-associated macrophages, but we won't get into those details here. It's not the purpose of this talk. Potentially, microglia fail to kill fungi efficiently, but likely they initiate local inflammatory responses. And this leads us to our future questions. What are these local inflammatory responses? What are the cytokines that microglia are doing? What cells are they recruiting? Do they increase vascular permeability? Is the blood-brain barrier function decreased? Is there an influence in the brain function in this area? Can it manipulate the neuroenvironment? And at the same time, from the other end, how does the fungus manipulate the cells around here to avoid killing? And can we harness this knowledge for therapies? Other research that we have going on is, again, to study how the macrophage and the Cryptococcus can adapt to infection. I would refer you to Clara Freitas, which is studying this, who just published a paper in a collaboration with Amelia Boca, just published a paper on metabolic adaptations during infection. I give you a summary of this. I give you a summary of all the different lovely images that we can generate with tissue clarification. It's a really important tool to study these interactions and others. So I encourage you to try it. And there's different methods of doing some of the methods really cheap, much cheaper than what we think. And I really thank the scientists and others, because when we first published this work, actually, we got a good amount of media attention. So if you want to get the gist and the summary of this and reactions from other people, I would direct you to this link, because we got featured. This work got featured on the scientist studying the brain using clarification. And that was a really, really nice. And, it's good to see that your work has a importance beyond, let's say, the university. And I remind you to always look at all the different perspectives, because it's possible that the different perspectives really inform the direction to go, and that you can find an explanation that is a lot more comprehensive and a lot more accurate once you consider all the other perspectives. And finally, I really like to thank Vanessa Francis. She's here. And she is the person that did most of the microscopy. She was the hands on the microscope. She's a wonderful microscopist photographer that was doing a postdoc with me. She's now moved on, but her work, she is really the person who did most of the work. I would also like to thank Will Newton, who's still continuing his PhD, and all of the collaborators, particularly for this work: Corin Little, Darren Thompson, Peter Cook providing us mice, John Whitten, who helped us with all the neuro stuff that we were new to, and who was really, really collaborators. So really huge thank you to everyone who's participated in this effort. It was started during COVID, so it was really a group effort in order to get this started. But then Vanessa came in and she swooped in and she knocked it off the park. And finally, thank you to all of my funders and the British Mycological Society. And I think I will stop sharing, if that's okay, Jan.
That's perfect. Thank you so much, Carolina. That has got to be one of the clearest talks that I've heard for a very long time. So thank you so much for making that so accessible. Really excellent. The microscopy, it was just phenomenal. Well, I've got a few questions. So if anybody else has got any questions, could people pop them in the chat? Oh, we have one already. So be polite, I will ask that first. So this is a question from James O'Connor, and he says, thank you, Carolina, a really interesting talk and great work you're undertaking. Is there anything that can be harnessed diagnostically from what you have discovered about the pathogenesis of Cryptococcus neoformans, for example, biomarkers or similar?
That is a really, really interesting question. Not yet, but for sure, right? I know that there are some people who try to find how many of us have been exposed to Cryptococcus, whether you can detect foci of trees to see if maybe there's more infection because there's a higher load. But I have to say, not yet, but work to be done.
Yeah. Thank you very much for that, James. Jane Haydecker from Kent says, fascinating talk. Thank you. If anybody else has got any questions, then please pop them in the chat now. So there's a couple of things that strike me about Cryptococcus neoformans, Carolina. One of them is a very basic question. That is that I don't really understand why it has such an affinity for the brain. Is there anything known about why this pathogen in particular seems to like to really colonize the brain out of all of the organs?
I mean, I think that is the crux of the question, right? We don't really know. We think partially, if you wanted to propose some explanations, partially would be it's a fungus that doesn't really like hypoxic environments, doesn't really grow very well. So the brain and the lung tend to have a good amount of oxygen, so that could be it. some people have posited that it could be because Cryptococcus could benefit from neurotransmitters, and that would facilitate some of its defense mechanisms. And this is dopamine and feeding into melanin. But we don't really know. But actually, I don't think we know that from the majority of the fungi. Can we really say why is Candida in the gut and in the vagina versus? It's just we know it's a feature of what environment they prefer, but it's also a feature of, for example, from the lung, we know that Cryptococcus can be disseminated throughout the entire body, and it lands where it lands. And this is kind of like the seed hypothesis in cancer, where the seed may go everywhere, but it will only grow if the soil is correct. And we know that the Kupffer cells in the liver and the spleen cells, the spleen monocytes, are a lot more effective in killing Cryptococcus. And that was work by Shi in Maryland, University of Maryland. So it does seem that in those two tissues, for some reason, the macrophages have a particular characteristic where they're effective at killing Cryptococcus, and you don't really have a lot of fungal load. You can detect it, but it doesn't grow. The ones in the brain don't kill it. The ones in the lung don't kill it. So then the fungi persists and continues to kill. The rest to be discovered.
Yeah, no, fascinating. I think the
Oh.
talked about that surrounds Cryptococcus, but also I know that they form these titan cells. And I was just wondering, using this fantastic microscopy, are you able to, see how frequent the cryptococci are forming titan cells, or how many of them are encapsulated, for example?
Absolutely. I will tell you, probably that was the second person I showed some of the images we had to, because when we first got into the microscope and we started having really clear images of the lungs of heavily infected mice, you know that in my office sits Liliane Caramera, who studied titan cells and who also studies titanization, and Liz Ballou. So I literally said, look, you need to look at this picture. We can now very easily measure titan cells in vivo. So one of the things we discovered is, for example, in the nose, there's a huge amount of titan cells and it's a really fast formation of titan cells. And Liz Ballou thinks, because she thinks she knows why they're induced so fast there, and I'll let her discuss that. She has a theory, but absolutely, it's really powerful for studying titan cells because of the numbers you have, but also because you have a better image and you know that you're capturing the entirety of the cell. So absolutely, and we're doing that work.
When you're so in your brain images, which were absolutely phenomenal, when you see the odd yeast that perhaps isn't next to the microglia, can you see whether that's encapsulated? Has it put on that invisibility cloak? I love that term. I mean, do you think, can it hide from the immune cells, or what do you think might be happening there?
So Cryptococcus always has a little bit of capsule, and then it just makes it thicker when it gets into the host. So the capsule is there for sure. We didn't stain for it, but we know for sure it's there. We think it helps. It definitely immunomodulates, but we haven't really studied whether it's more in microglia than in other immune cells. So we think that it absolutely helps. I also know that there are people who are studying other cells in the blood-brain barrier. And I think we are going to need the entire field to study this, right? I know Rachel Nanganabisi, a collaborator, and Rebecca, also a good friend, Rebecca Drummond at Birmingham, is also studying different aspects, different time points, different cell types, to really start to get at all the aspects of this. What the capsule is doing there, for example, I'm referring to everyone because I do think we need to work as a field together. Luis Martinez in Florida is studying the capsule, and he really does help potentiate the growth of crypto. So if you isolate capsule and you administer it to animals, you're going to potentiate the infection. Even isolated capsule, you're going to potentiate infection. So a lot to do. It is definitely, the capsule is definitely immunomodulatory, but I'm also sure that it's doing secreting other immunomodulatory proteins.
No, it's absolutely fascinating. And I think that having being able to actually visualize the whole process is such a huge strength that you're right. I think that the whole field could benefit from not just looking at Cryptococcus neoformans, but looking at how candida's disseminating,
Yeah.
from the gut. What frequency does that happen, rather than just injecting candida into the tail vein? It's not
Yeah.
realistic, is it? So, no, I think it's an absolutely fascinating model, and it's really, really excellent research. So thank you very much for that.
Thank you. Thank you.
So there were no further questions in the chat. So rather than me bombard you with other questions, I think I will finish by thanking you very much for, again, a very, very clear talk. That was absolutely amazing. I'd like to thank the audience as well. And I will bring this evening to a close. So it's goodbye from me. And Carolina, if you would like to say a couple of words.
Just a huge thank you to the BMS, to you for hosting, and to all the staff in the background, administering everything in the BMS, and to everyone who has helped us along the way. And also, again, to all the collaborators who made this work happen, really.
Okay. Well, thank you, everybody. Have a good evening.
Thank you. Have a good evening. Bye-bye.